Bueno, vamos a empezar. Buenas tardes a todos, gracias por su presencia, a los que estamos aquí en el auditorio y a los que nos siguen en línea a través de Zoom y YouTube. Realmente es un gusto presentarles al doctor Sriram Chandrasekaran. Él es Assistant Professor de Biomedicina y Ingeniería en la Universidad de Michigan and Arbor, donde dirige el Laboratorio de System Biology and Drug Discovery. El doctor Chandrasekaran recibió su doctorado en biofísica de la Universidad de Illinois at Urbana-Champaign y después trabajó con una beca de Harvard en la Universidad de Harvard y en el MIT al mismo tiempo. El doctor Chandrasekaran ha recibido diferentes premios a su trayectoria académica. Entre ellos está que recibió una beca predoctoral del Howard Hughes Medical Institute, después una beca de postdoctorado en Harvard. Recibió el reconocimiento a los investigadores más sobresalientes menores de 35 años por el MIT. Y después también tiene un reconocimiento, un premio de Ike Cobra Society a jóvenes investigadores. Recibió también el premio Mira del NIH y de la Fundación Dreyfus, Camilla and Henry Dreyfus recibió el premio de Machine Learning in the Chemical Sciences. También fue orador invitado al Fórum de Líderes Emergentes del 2022 por la National Academy of Medicine. El doctor Sriram, como ustedes vieron en su página, trabaja en varios aspectos incorporando machine learning y modelos matemáticos a predecir, descubrir nuevas drogas para tratar diferentes enfermedades. Realmente le agradezco a Sriram haber aceptado la invitación. Thank you very much for accepting the invitation for the second time and share your knowledge with the students. And thank you very much for being here.
Can everyone hear me? Hear me okay? Great. Well, thank you so much for the kind invitation. It was really nice meeting everyone. Again, for the second time, it's been 5 years since I came here. So I really enjoyed interacting with all the students. And even this time, I allowed giving the guest lecture and really enjoyed talking to you guys. So once again, feel free to stop me anytime if you have any questions. I wanted this to be informal as possible. So if you don't understand anything, please stop. So today I'm going to talk about some of the machine learning methods my lab has developed for drug discovery and drug repurposing. So one of the main focus for my talk is going to be on antibiotic-resistant bacteria. So as some of you might know, there's one of the major global problems right now is antibiotic resistance and the spread of drug-resistant pathogens. And the world is running out of new antibiotics to treat drug-resistant bacteria. So today I'm going to talk about some of the new drug discovery methods my lab is developing to address this key challenge. So how did we get here? So antibiotics is, according to me, one of the most greatest discoveries of mankind. Right? Before antibiotics were discovered, 5 out of every 1,000 women who died during childbirth, and even a simple wound or scratch can become, life-threatening. People might lose an arm because of a simple scratch. So that was before antibiotics were discovered. Now we don't have to, go through all these, but unfortunately, due to antibiotic resistance occurring, you may have to go back to the era where, simple wound or scratch could become life-threatening again. So one main reason for antibiotic resistance is simple evolution. This plot here is showing data for one organism, gonorrhea, but it applies for other infections as well. Antibiotics were introduced around the 1940s, year of introduction here, and this is the year in which resistance was reported. And you can see that every time a new antibiotic was introduced, within a few years, people discovered drug-resistant bacteria. Right, in 1935, 1940, then penicillins were discovered, drug resistance for penicillins were discovered in 1960, and then we discovered other tetracyclines, and then we discovered drug resistance for those, and then we kept on discovering new antibiotics, and then we kept on discovering new drug-resistant bacteria, right? So this is fine as long as we keep discovering new types of antibiotics. But unfortunately, that's not the case. During the 1940s and 60s, we discovered a lot of new antibiotics. But more recently, there's hardly been any new antibiotics that have been discovered. So coupled with the last slide where we've been identifying drug-resistant bacteria, and here we hardly have any new antibiotics that are being discovered. So it sort of caused the perfect storm where we are experiencing drug resistance on one side and lack of antibiotics on the other. And one promising solution is sort of inspired from Batman movies. In a very old Batman movie around 2000, Joker, tries to kill people. And what he does is that he poisons a lot of household products, right? Like hairspray, toothpaste, he poisons a little bit of everything. But the interesting thing is that, if you use toothpaste alone, you don't die. Only if you use, for example, the red colored products like toothpaste with makeup and hairspray, only those people, suffer from the poison. Similarly, if you take only the green products, then you die. But if you mix other things or use one thing at a time, then you don't die. So he sort of discovered combination therapy. What he found was that if you use one product alone, it's not toxic, but together they become synergistic. And it targets certain type of people who use 3 products at the same time, right? So that's the idea we are going to use here too, is that of combination therapy, where you combine multiple drugs at the same time. And just like what Joker did, by combining drugs, you can enhance their potency. And by taking non-toxic compounds, you can combine them to make really potent antibiotics. And another thing is you can also reduce resistance. The probability of a bacteria evolving resistance to multiple drugs at the same time is much lower than in each individual drug. So you can reduce resistance, and you can also modulate the spectrum. You can target multiple bacteria at the same time, or you can have a narrow focus and focus on a certain strains of bacteria by changing the combinations. And then another key aspect here is what we call repurposing. So you can take drugs that are being used for other diseases like malaria or cancer, and then you can repurpose them and use it for antibiotic discovery. So that reduces the cost for antibiotics, but also time involved in reaching the clinic. So repurposing has a lot of advantages, and using combinations, we can repurpose drugs faster. So, but there are also challenges in designing drug combinations. One main challenge everyone talks about is that there are a large number of combinations, right? For example, if you just want to design a 4-drug combination that's currently being used for treating tuberculosis from just 100 drugs, that requires screening like 3.9 million combinations, right? And if there are more drugs, then it explodes exponentially. And so you can't do this experimentally, so you need computational tools to identify the most effective drug combinations. And there are also other challenges that I'm talking about today. So in addition to a large number of combinations, we actually don't have data to build, like computer models for many pathogens. There's E. coli is very well studied, but other organisms don't have enough data to build these models. Another related challenge is that the environment in which the bacteria is going in, it could be the immune environment or the metabolic environment. They're very different from the lab where you grow the bacteria in a petri dish versus inside a lung or liver where the infection is happening. The environment is very, very different. And so we are developing models that capture the environment in which the bacteria is going in computationally. So my lab takes a systems biology approach for drug discovery. So traditionally, drug discovery was more target-based. They will find an enzyme that's important for bacteria and then try to design a drug that blocks that enzyme. Whereas now people understand that we need to take a more holistic approach where you need to take into account the properties of the pathogen, but also its interaction with the host immunity, the environment in which the bacteria is growing in. So all these needs to be taken into account to design more effective drugs. And so my lab's been developing sort of what we call as a hybrid AI methods. So we use machine learning for drug discovery, but traditional machine learning is what we call as a black box. You really don't understand how the machine learning algorithm works. So we are sort of combining machine learning methods with more mechanistic engineering-based, biochemistry-based models where you understand what's going on in the model. We know what genes or what pathways are involved. We can sort of understand the underlying mechanisms in these models. So we are combining these two to sort of get the best of both worlds. We can get the predictive power of machine learning, but also the mechanism of understanding that you can get from more traditional engineering models. So we've developed a range of methods using this hybrid AI. So that I'll talk about today. We've developed methods called Indigo, Magenta, and Caramel that address many of the limitations, challenges that I talked about earlier, right? And they help to, like prioritize experiments that are costly and expensive. And also another cool concept that I'll talk about today is what's called transfer learning, where we can take models that we've built for like E. coli and then apply it to less studied pathogens like tuberculosis or Staph aureus. So I'll jump in briefly to describe how Indigo works. Indigo was the first method we developed. And so the underlying idea behind Indigo is that it connects genes that respond to certain drugs and uses that information to predict how two drugs are synergistic or antagonistic. So just to give you a better example, everyone knows Netflix, right? Netflix has a very good movie recommendation algorithm, right? And the way it works is that it uses two sources of information, right? One is your past history of what movies you watched, and the other information it uses is the properties of the movies, right? If you had watched movies by one actor, then it looks at the properties of those movies and then recommends more movies by that actor or director. But if you watch comedy, then it recommends more comedy to you, right? So it's two sources of information. Similarly, that's how Indigo works. Indigo takes in two types of information. One is known examples of drugs that are synergistic or antagonistic, that's well known. And then the second information is the property of the drugs. Just like the property of the movies, like comedy or romance, similarly, the drugs have properties, right? They could target the cell wall or ribosome or other processes. So it takes in the properties of the drugs and then known examples of drugs that are synergistic, and then it makes new predictions. For unseen drugs, it can recommend drug C and D might be synergistic or antagonistic. So that's how Indigo works. And it uses an underlying machine learning algorithm called random forest that builds decision trees like this that connects genes that respond to a drug. And based on that information, it can predict, like synergy antagonism for new drugs. Yeah, so just to describe it in more detail, like the underlying mechanism behind how Indigo works is that it builds what's called as a drug-gene interaction network. So it uses data from transcriptomics or chemogenomics data where, for example, you expose a drug to a bacteria like E. coli, and then let's say you delete a gene or mutate a gene in that bacteria, right? So some genes, when deleted, can enhance the potency of the drug, or some genes can confer resistance. The bacteria might grow faster when that gene is deleted in presence of the drug. So all this data goes in and forms what's called the drug-gene interaction network. So out of maybe 4,000 genes, there might be 20 genes that increase sensitivity to a drug, and there may be 20 that confer resistance. So we can build a drug-gene interaction network that's unique for drug A, and drug B, which might have a different mechanism, will have a different drug-gene interaction network. So these are the properties that Indigo uses, to predict new drug interactions. So it uses this data on drug-gene network, and then uses that to predict how two drugs, let's say drug A and B, interact with each other. So this idea takes a more systems approach. So rather than just focusing on just the target of the drug, like its cell wall or ribosome, we take a more holistic approach where we take on all the processes that a drug is involved in. There could be processes that are involved in uptake of the drug, involved in drug metabolism, secondary processes like stress, oxidative stress that are involved, all together influence how potent an antibiotic is and whether a cell will die or survive an antibiotic treatment. So we take the systems approach to predict the potency of a drug. So that's the theory. So I'm going to give you examples of how Indigo is performed in actual experiments. So originally what we did was we did a training set, just like I mentioned for Netflix, we needed a training example, right? So we measured interactions between various antibiotics. I'm showing here this experimental data. These are like, erythromycin, tetracycline. These are antibiotics that target different cellular processes. And then we measured whether these antibiotics are synergistic, shown in blue, or antagonistic, shown in red. Right? These are drugs with different mechanisms. And one thing you can see is that there's more antagonism than synergy. So just sort of random, if you combine antibiotics, they're usually going to be antagonistic. And then using this training data, we connect that with the properties of these drugs using what's called as chemogenomics data for a lot of other drugs too. So we have around 20 drugs here And then we are able to now connect that to the chemogenomics data, which we have for hundreds of other drugs, right? So now we can make predictions for all possible combinations for which we have data for. So in this case, we did pairwise combinations first. So we had like around 50,000 combinations that Indigo predicted. And then we experimentally tested the most synergistic and antagonistic ones. So we picked drugs that were completely new, that Indigo had not seen before, that had new mechanisms of action. So it's sort of like a worst case scenario of testing Indigo. So we picked drugs like the target RNA synthesis, which was not in the training set. And then we experimentally measured interactions between these new drugs and the drugs in the training data experimentally. And then we compared Indigo's predictions with experimental data. And what we found was that around 7 out of 10 predictions matched experimentally, so around 70% was accurate, which is much better than random guess. And the cool thing was that it was able to accurately predict synergy, which I think was what everyone cares about, is that synergistic interactions were significantly different from the antagonistic prediction. So Indigo is able to separate out synergy from the rest of the data with a significant p-value. So this showed that Indigo works really well. It's able to identify synergistic interactions from the data. Next thing we did was we looked at 3 drug combinations. Can we go from pairwise to 3-way, which is even more computationally expensive? And then we picked, once again, new drugs that Indigo had not seen before. And then we did all 3-way interactions between them. So this is actually the experimental data. And what we found, read this antagonism. And what we found was that when you go up higher order, like 3-way or 4-way, antagonism dominates synergy. So we actually see more antagonism here and very few synergies shown in green. Just actually a couple of synergies are found when you go higher order, right? And once again, we're able to predict synergistic interactions really well compared from the rest of the data. So I would say that the predictions are not perfect, right? It's good, it's statistically significant, but it's not, we're not able to exactly predict every single synergy. And so we went back and looked into the model, see where it fails. Like what are the cases where it does really well? What are the cases where it doesn't do well? And one thing we found was that the error in the Indigo predictions correlates with the chemical properties of the drugs. So if a drug is very hydrophobic, then it's really hard to predict, right? And then some of these chemical properties correlates with the error in the model. For example, you can see here, really large molecules, actually we can predict really well, but if it's really small, it's hard to predict. The reason we think is that these small molecules can just diffuse into the cell and might have a lot of promiscuous interactions, and that's really hard to predict in the model. Similarly, if the molecule is, is hydrophobic, then it's really hard to predict. Because hydrophobic molecules could bind once again to, like hydrophobic pockets or the membrane. So that once again makes it really hard to predict. So so the models are perfect, but maybe for the next iteration, we can include these properties to further improve the model. Any questions so far? Right. So just to summarize the first quarter of the talk, I talked about the challenge of antibiotic resistance, and then I talked about how combination therapy can address the challenge of drug resistance, and talked about how Indigo can screen through a large number of combinations, right? The next part I want to talk about is, how can we apply this data we generated for E. coli to other clinically relevant pathogens like, Staph aureus, right? Where that's where you need treatments for, but there's no data to build these models. So that's the next challenge I'm going to talk about. So we've been developing what's a type of machine learning called transfer learning, where you can take models that you build for one system and use it for other. Like in Google uses this for like it can learn images from like stop signs or traffic, and then you can take the same model and then apply it to identify different types of cats. So it's same image processing algorithm. It's just using it for slightly different tasks, and that's what we have been trying to do is develop models. We're using models that we develop for E. coli and then apply it to other organisms. And the reason we can do this is because, as I mentioned, we are developing more mechanistic models. So these are based on known biochemical interactions, so we can modify them for different organisms. So if you remember, I mentioned that we build these drug-gene interaction networks and use that to predict drug-drug interactions, right? And we've done this for E. coli, and what we know from genetics is that we know genome sequence for many of the pathogens, and we know what genes are conserved or not conserved. So we call it orthologs, that they are conserved between species, And we can take this E. coli model, and then we can match orthologs and sort of get a similar model for another organism. So for example, if gene 1 and 2 are also present, let's say it's Staph aureus, are present in both E. coli and Staph aureus, so those interactions remain, right? If gene 3 is not present, then that interaction will be removed. And then if you go into a machine learning model, then we can see that, for example, for some combinations that rely on gene 1 and 2, right, that interaction remains the same. So these drugs that depend on gene 1 and 2 alone, they will still be synergistic, right? But if some combinations rely on gene 3 that's not conserved, then this interaction is going to change, right? So this interaction will not be conserved. This will be conserved based on the underlying genes. So we can use this idea to now predict, make predictions for another organism. So we tried this for Staph aureus, which is more clinically relevant than E. coli. So we first experimentally measured drug interactions in Staph aureus. Right, you can see, once again, red is antagonism, blue is synergy. You can see more antagonism here than synergy. Once again, if you combine antibiotics randomly, they usually end up interfering with their action. But the cool thing is that now we can actually use Indigo for the transfer learning approach to predict synergistic interactions and separate them from antagonistic ones with surprisingly good accuracy. And the cool thing about this is that we did not use any data for Staph aureus. This model was built for E. coli, just using the DNA sequence, which is easily available. We're able to predict drug interactions for another species, which might be more clinically relevant, using E. coli data. We also applied this to tuberculosis, Mycobacterium tuberculosis, which has a huge burden worldwide. It leads to more than 1 million deaths annually, and the treatment takes a lot of time, from 6 months to 2 years for drug-resistant TB. And so we built a similar model for Mycobacterium tuberculosis. And in this case, we actually built a bigger model. We collected more data compared to the E. coli study. So we had more than 200 drugs for the tuberculosis model. And tuberculosis is normally treated with more than 2 drugs. People give 4 drugs, sometimes 5, even 6 drugs if it's drug resistant. And so we tried to look for all possible combinations of these 200 drugs. We got around 5 million combinations we predicted computationally. And then we tried to identify combinations that do better than what's being used clinically. You can see that clinically used combinations are mildly synergistic. And then shown here on the left, any randomly chosen combination is more antagonistic, like we saw in E. coli and Staph aureus. If you just combine drugs at random, they usually end up being antagonistic. But we did find some combinations that are more synergistic than being used clinically. Then we collaborated with David Sherman's lab at UW, where they have a BSL-3 facility where they can actually grow tuberculosis bacteria and then test these predictions. And so we tested a few synergistic and antagonistic ones, and we found a very good correlation between like what Indigo predicted to be synergistic and what was experimentally measured by their lab. We got a very good correlation of more than 0.65 for around 32 2-way and 3-way drug combinations. So some of them have 3 drugs, some of them have 2 drugs here. And I want to highlight one cool combination that we found. This is actually 2 drugs that are not currently used for treating tuberculosis. One is chlorpromazine, which is actually an antipsychotic drug, right? It's used for a completely different indication. And spectinomycin is an antibiotic, but it's not used for other infections. And what we found was that, for example, this is an experimental assay. You can see that spectinomycin at roughly 2x its dose kills all the bacteria. Chlorpromazine 2 at around 1x its dose kills all the bacteria. But together, 1/16 of spectinomycin and 1/16 of chlorpromazine is enough to kill all the bacteria. So that's an example of how we can find highly synergistic combinations by this one reduces the dose for both the drugs and also enhances their potency. So this is one example of drug repurposing where you're using drugs that are used for something else that could be then used for treating tuberculosis. Another interesting thing we can do with since a mechanistic machine learning approach is that we can also identify what genes are important for affecting synergy, right? We can go back into the model and see what genes the model used for predicting synergy for different drug combinations. So we did a in silico screen sort of to find specifically what's called as transcription factors that seem to influence drug interaction between various antibiotic combinations. And we found one top regulator, RV1353C. So this is not studied. So this gene doesn't have a name. That's why they have this number here. But what we found was that this gene seems to influence the drug interaction scores for many drugs. And you can see in the bottom here that we screened a lot of antibiotic combinations. And when we deleted this gene, it made some drugs more synergistic, especially those that involves like bedaquiline here, streptomycin, that are currently being used for treating tuberculosis. And so we went back into the lab in Shah and we tested these predictions. So when we deleted this gene, it made some combinations more antagonistic, but when we overexpressed it, it made them more synergistic in every single case. And this drug was a control. It had no impact, and you can see there's no impact for this one. So using this now, we can also identify what genes regulate synergy antagonism. And then maybe, there could be a fourth drug we could design that blocks or overexpresses this drug, and that can make some drug combinations more synergistic. So we can engineer synergy rationally using this method. So just to summarize this part of the talk, I mentioned about the lack of data for building these machine learning models. And I talked about transfer learning, where we can take these models that we built for E. coli and we can apply it to Staph aureus. And I also talked about how we can use this for tuberculosis and screen large number of combinations. For the next half of the talk, I'll talk about how we can better capture the in vivo environment, like the metabolic environment and the immune environment in an infection site. So first thing I want to talk about is metabolism, right? So now, normally, we think of antibiotics as blocking a certain key molecule, and that's how cell death happens. But people have recently found that the presence of metabolites can completely change the potency of antibiotics. So here was a really cool study where they took this antibiotic called kanamycin to a bacteria that is actually resistant to kanamycin, right? So when they added kanamycin to this bacteria, the cell didn't die. The cell was growing happily. But then they added kanamycin with glucose or alanine, right? And then the cells died, right? These are the same resistant cells, but just by adding these metabolites, you can completely change the potency of this drug. Right? And people have found other examples for other antibiotics as well. And so this is important now because inside your body, infection site, there are a lot of metabolic changes that happen during an infection, and adding antibiotics can also produce different metabolites. And so, the bacteria doesn't reside on empty space. There's a lot of metabolites that are coming in and going. There's a lot of metabolic interactions with the immune cells. And so we need to take into account the metabolic environment when we're designing new drugs. So to do this, we've developed a new method called Magenta, which is like an improved version of Indigo. So Indigo takes in this part, right? We have the drug-gene interactions, and then it predicts drug-drug interactions, right? So what Magenta does is that it also takes an additional input, which is the metabolic environment in which the bacteria is growing in. So rather than just saying drug A and C are synergistic, Magenta will predict a condition-specific interaction. It's a drug A and C are synergistic in the presence of glucose, or it will predict drug A and B are antagonistic when glycerol is present. So it makes a more nuanced prediction. The way it's able to do that is that it also takes into account genes that are important for metabolizing certain metabolites like glucose or glycerol. So glycerol requires maybe gene 7 and 2. So it takes this data into account and into the same machine learning model, and it makes more condition-specific predictions. And so just to give you an example, we tried this in E. coli too, but I'm just showing data for Acetobacter, which is a very important pathogen that is commonly seen in hospitals. And it's actually a high priority pathogen for the US CDC and WHO because there's hardly any drugs for treating this pathogen. Right? And so what we did was we took this bacteria and then we grew it in 3 different conditions. One is the standard lab condition, LB media. Another one is like minimal media with glucose. Another one is minimal media with glycerol, right? And then we measured the synergy and antagonism for different drug combinations shown here. So one thing you will immediately see is that depending on the metabolic environment, the drug interaction scores change a lot, right? Some drugs that are highly synergistic here, like ampicillin and amikacin, are slightly antagonistic in LB media, or neutral in glycerol. So depending on the environment they're growing in, the synergy and antagonism can actually change, right? But the cool thing is that we're using Magenta, we're able to actually predict how the interaction scores will change in different media conditions. So we can predict interaction scores in LB media also in glycerol and glucose. So each of these conditions are able to accurately predict the synergy or antagonistic outcomes. We did the same thing in E. coli, and we got even a better, higher correlation. So we've also been slowly doing similar stuff for cancer. Today, I'm going to primarily talk about infectious disease, but we've also been doing similar work where we're trying to understand how the metabolic environment changes the potency of anti-cancer drugs. And in recent study, what we did was we took the same cancer cells, added the same drug, but we added different metabolites to it. And what we found was that the metabolic environment in which the cells are growing in has a big impact on the sensitivity to different drugs. So in this case, for this epigenetic drug, vorinostat, the potency changes a lot depending on what metabolite is present in the environment. Just like the bacteria case, the environment has a huge impact. Metabolic environment has a big impact on the potency of different drugs. And we are actually able to identify metabolites like the ones I'm showing here that are highly synergistic with specific epigenetic drugs. Like for example, we found sorbitol was synergistic with this drug, GSKJ4. Hydroxyglutide was synergistic with vorinostat and so on. So we have this drug metabolite interaction network that we have identified for epigenetic drugs. So a related challenge when I talk about metabolism is heterogeneity, which is that, if you take the same cells in the same condition, for example, even the same condition, some of the cells might have different metabolic activities. Some cells might respire slowly, some might have high glycolysis, just sort of random noise, right? That's we call as metabolic heterogeneity. So that happens even in the same condition, right? And so when you add an antibiotic to these heterogeneous cells, some bacteria might actually survive because they have a unique metabolic state, right? They might be having low oxygen levels or low glycolytic activity. Some unique metabolic state helps them survive antibiotic treatment. And then when you remove the antibiotic, these cells grow back, to give the whole population. So it's not genetic, but it's just metabolic. They just have a unique metabolic state that helps them survive these. So these are called quiescent cells or tolerant cells. They have different names in literature. And so we want to capture this metabolic heterogeneity in our computational model. And so very recently we developed a method called Caramel that accounts for this heterogeneity. So what it does is that it creates computationally a unique population of cells, each with its own unique metabolic activity. And then it then exposes these cells to different antibiotics. And one thing we found, for example, was that when you add combinations of drugs to in our virtual model, we find that there are different subpopulations. In this case, this drug combination, azithromycin with minocycline, is actually predicted to be a neutral or non-interacting in our Indigo and Magenta model. But what Caramel model found was that there are actually two subpopulations. One population, this drug combination is slightly synergistic, and another population is slightly antagonistic. So together, in the bulk Indigo model, it appears to be neutral, but then in reality, there's actually two subpopulations where it's slightly synergistic and antagonistic. And we have made predictions for a lot of other combinations as well. And so what Caramel does is that, it's a generalizable approach where it generates a population of cells, and then it can now predict what happens when you add drugs together, but also what happens when you temporarily separate drugs. Let's say you add drug A first, wait for a few hours, and then add drug B. It can predict these sequential interactions. And we're trying to use Caramel to design these cyclic protocols where you add drug A, which kills, let's say, some subpopulation of cells, some survive. Then you find another drug B that kills another subpopulation. Then maybe you can add drug A again or a different drug C that eliminates different subpopulations. So then that reduces the chance of developing drug resistance. And so the underlying idea is that we use these detailed metabolic maps that are available for various organisms. These are called genome scale metabolic models. And what they do have is that they catalog the interactions between what metabolites are involved in what reactions, what genes encode these enzymes, how these reactions are organized into pathways, and how they fit into the whole network. So we feed in single cell proteomics data into these models, and then we can generate a population of cells with different metabolic state. And so that's the overall idea behind Caramel, is that we take different omics data, and then we can integrate these with these metabolic models. So it could be proteomics data we can incorporate transcriptomics or metabolomics data, and then we can convert these data into a single format, right? We can using these models, and then we feed these output of these models into machine learning algorithm, and we can use this to predict, synergy or sequential synergy where you separate drugs over time, and we can predict cyclic combinations of drugs using this. We can also simulate our own media condition now. Like for example, we can simulate glucose and glycerol together or any other complex media. We can use these models to simulate different metabolic environments, and we can predict the impact of both the environment, time in a single algorithm. So the last few minutes, I want to talk about the last part, that is how to better capture the in vivo environment, especially the pathogen immune interactions. I think this will also be a nice segue for your next week's lecture. I know next speaker is Dr. Judy Lieberman, and this was a collaboration with Dr. Judy Lieberman's lab, where we tried to study how a specific type of immune cell called a killer T cell eliminates different pathogens. Right. So many of the pathogens I talked about today, like E. coli, mycobacteria, they're all what's called as intracellular pathogen. And so the immune system uses killer T cells to eliminate intracellular pathogens. And the way it does that is by producing a key enzyme called granzyme B. So granzyme B is a protease that goes in and cuts proteins in like the target bacteria, like in E. coli or tuberculosis, and that's how it causes cell death. And so what we're interested in is that we wanted to know how exactly granzyme or killer T cells eliminate diverse pathogens. The same enzyme is able to, kill E. coli, tuberculosis, listeria. In reality, if you use antibiotics, you use separate antibiotics for listeria, a different one for tuberculosis or E. coli. But the same immune enzyme is able to kill diverse bacteria on its own. So we wanted to know how it does that. Like, does it go after a specific protein like an antibiotic, or does it just randomly cut proteins? And if it goes after a specific process, is it known targets, or is it something new that we don't know about? And by studying this, we can design better drugs that work in synergy with the immune system. So maybe we can enhance the immune activity or find drugs that complement the immune function. To do this, we collaborated with Judy Lieberman's lab. We looked at 3 pathogens, E. coli, mycobacterium, tuberculosis, and listeria. And then what we did was we exposed proteins from these bacteria to granzyme B. So granzyme B, as I said, is a protease, so it cuts proteins. So now if a protein is targeted by granzyme B, and then you use gel electrophoresis or mass spectrometry, the weight of the protein will change, right, before and after treatment. So by looking at the change in the mass of these proteins, we can identify which proteins are being targeted by granzyme B in all 3 bacteria. And so we put this on a metabolic map I'm showing here, is that you can see a lot of proteins are being targeted. So shown in red are the ones that are shared, cut in more than one species. And you can see that proteins in TCA cycle, glycolysis, oxidative phosphorylation are all being targeted. So it looks random, right? But what we found is that when we analyzed this on a metabolic map, is that first thing was that these proteins are highly conserved. What we found was that when we looked at more than 317 bacteria genomes, or actually 317 organisms, we found that all these granzyme targets were highly conserved. So what this means is the same enzyme is able to kill diverse types of bacteria, or even like other types of pathogens. So it's because it's going after highly conserved proteins. And another thing we found was that when we used this metabolic model to simulate what happens when these proteins are being cut, was that we found the bacteria was unable to grow in any of the metabolic conditions we tested. So for example, if granzyme B only targeted glycolysis, then the bacteria could have used like fatty acid metabolism to survive. But granzyme B is also targeting fatty acid metabolism. So maybe then it will survive using nucleotides, but nucleotides is also being targeted. So it's sort of cutting off all the backup routes the cell has to survive in vivo. And so when we did evolution experiments where you expose E. coli to a drug, or in this case granzyme B, we found that the bacteria was not able to survive even after many passages. If you did the same thing with an antibiotic, you'll see that after 4 or 5 passages, this growth of the bacteria will go up over time because it's evolving resistance. Whereas with granzyme B, it was not able to evolve any resistance because it's using this combinational approach by targeting multiple enzymes at the same time. So another new direction we're also going is that we are trying to model the entire infection site, in this case for tuberculosis. So what happens is that when most of these respiratory pathogens, when they go into the lung, they don't just stay there. They are surrounded by many immune cells, like macrophages and others. And so they form a ball around the pathogen. And so in the lab, when you add a drug to a bacteria, you just add the drug and the drug enters the bacteria. Whereas inside the body, the drug has to diffuse, through the bloodstream, into the lung, through these immune cells, and then finally hit the bacteria. So it's a big diffusion biophysical barrier, right? So some drugs can easily diffuse in, some cannot. So we want to take those properties into account. Some drugs might also, have different pharmacokinetics where they can diffuse in, but also leave the site fast, quickly. So we want to take that dynamics into account. And so what we did was we combined our machine learning model with a more with a chemical engineering type of model to understand, like how synergy that's predicted by Indigo actually pans out inside the infection site. So we used a method called GrandSim that predicts how well drugs diffuse into the site of the infection. So what we get as an output is that, for example, in this case, this is the infection site. You have bacteria inside, macrophages, immune cells outside. And we can see that for this drug, shown its concentration levels, it doesn't actually reach the site where the bacteria is. Right? It's sort of staying outside, but it doesn't actually go in. Right? So even though Indigo might predict that this drug is highly synergistic, it doesn't actually go reach the bacteria where the bacteria resides. Right? And so when you calculate the interaction score now, the model predict now antagonistic interaction because the drug is not actually even going in. So its potency is much lower, right, where the bacteria is. So using this approach now, we can predict what's called as an in vivo potency. So what Indigo predicts is sort of an in vitro, the lab potency. And by calculating this pharmacokinetic interaction, we can calculate an in vivo potency in our model. And so we've done this for a lot of drugs. And cool thing is we can go back to existing clinical trials that have been done, that have been tested in patients for different drugs and drug combinations. And then we can use our this entire approach to rank different drugs, right? Okay, this drug regimen would do well, this one will not do well, right? And then you can go back and see in clinical trials whether they actually did well or not. And so each dot here, you can't read it, but each dot here is a different clinical trial of a drug combination. So some involve completely new drugs, others are the same drug combination, but they're dosed differently. Some drugs are given more every day, or others are given like twice a day. So the dosing changes, and we can predict how well the drug combinations are potent in vivo using our method. And then you can see that the ones that we predict to be highly potent in vivo are also the ones that are, like highly efficacious in clinical trials. So what we're doing now is we're trying to predict new drug combinations that are somewhere here that are better than what's being used clinically. So we can, then use those for future clinical trials, or identify new, more potent drugs for tuberculosis. All right. So the last few minutes, I want to talk about where we are moving towards in the future. Now we want to try to build more personalized models. Each person has their own unique immune system. Their pathogen might have a unique genetic makeup, right? And so we want to come up with more personalized models that take into account the individual's properties, the pathogen's properties, come with more personalized prediction. And so to do this, we've been developing a machine learning model that uses data from like patients' health records, like their BMI, their smokers or non-smokers, that kind of patient level data, and also data on the pathogens, like what the DNA sequence of the pathogen is, and then properties of the drugs. And also we have data on like chest x-rays, CT scans from patients. We are trying to put them all together in a single computational model and then predict, like whether a patient will survive or not based on the drugs they were given. Right? So now we can use this method to maybe try out in silico what drugs will really work for a patient or not before actually giving it to them. So we can, we've done this for tuberculosis, where we have worldwide data from various countries like Europe, Asia, and Africa. And then we have collected all these data on the patient properties, pathogen properties, and then we built a single machine learning model that can predict whether a patient will survive a treatment or die. And so for each patient, we get an output something like this, where, in this case, the patient was predicted to survive. And it also explains why it made that prediction. So in this case, the age of the patient is very young. They're just 20 years old. They're from a country that is favorable outcomes, and their gender is male. The drugs that were given to them were very favorable, and the BMI is favorable. So all these are predicting that the patient will survive this treatment. But there might be cases where, factors that favor failure, like resistance, might predispose patients to failure. So for each patient, we can then go back and see why the model made that prediction. So just to summarize what I've talked to you today, I talked about antibiotic resistance. I talked about various algorithms my lab's developing, like Indigo for screening large number of combinations. I talked about transfer learning idea for applying to other organisms like Staph aureus, and then how we can simulate the metabolic environment and metabolic heterogeneity using Magenta and Caramel, and then how to simulate the infection immune site using methods like Indigo Grantham, and then we are moving towards more like personalized models. So yeah, with that, I would like to thank various collaborators that have helped me along the way, and various students in my lab, and funding sources, and thank you all for your attention.
Vamos a empezar la sesión de preguntas. Las personas que están en Zoom pueden escribir sus preguntas en el chat, igual a las personas que están en YouTube. ¿Preguntas aquí?
Well, thanks. I have two questions. The first one is about metagenomics. Could you use the data from metagenomics or of a specific sample to predict in softwares like Indigo?
I think so. Yeah, I mean, if you have the whole genome, I think if you do 16S metagenomics, that's only identifying the organism, right? In that case, then we have to go back to a database and see if there is a whole genome sequence for the organism. Then we can use that in Indigo, right? So for Indigo, we need to find the gene orthologs. If either you sequence the whole organism's genome, or if you have information, if you identify that organism, then we can, go back, match the orthologs, and we can use Indigo. Yeah, so it depends on if it's a completely new bacteria that no one has seen before, and there's no genome sequence, then it's hard. But if you know the genome sequence, then we can use Indigo.
Okay, thanks. Yeah. And the second one is, could you, I don't know how to say, could you applicate or, these technologies to other drugs resistance and other drugs like antimycotics or antivirals with the data of these pathogens?
Wait, are you asking, can we combine or can we predict?
Predict.
For antiviral, we haven't applied antiviral, but we are doing antifungal. We are building similar models for fungi, like yeast, but we haven't applied it for viruses. Is that what you're asking?
Like I don't know. Like in the case of viruses, can we predict with these technologies the different combinations?
For viruses. Okay, you're asking if you can use this for viruses, right? Yeah. I mean, in theory, yes, we haven't done so, but in theory, it should be possible, but we haven't done that. Yeah. That's but theoretically, it should be possible. The same concept should work. Yeah.
Thank you. It was a very interesting talk. I'm wondering, I guess at the end of the day, this is just like a game of numbers, right? As you show with the granzymes, if you have enough metabolic pathways that are blocked, then the bacteria will eventually die.
Yes.
I was in a talk talking about cancer that had a very similar concept. And there, the people said that you need to hit at least 5 different orthogonal mechanisms to really make sure that there's no way, just statistically possible that the cancer will come around. So would you have an approximate number of how many drugs would you need to really make sure that there's no chance, or the chance is astronomically small, that bacteria will ever come back from that?
Yeah, I mean, as you said, I've seen numbers. People say 6 is a good number, but I think it's also because it's the number of targets it's targeting. So granzyme is technically one molecule that's hitting everything. So I mean, there are antibiotics that trigger 2 different processes that they block too. So if it's possible that if you like design molecules that activate the immune system and also block the bacteria, like that's 2 different modes of action. So I think, maybe if you target more than 2, I think that's already a good chance it will reduce resistance. Yeah, but I don't know what the exact number is, but people have said 6. I've seen theoretical studies that have seen 6. I think it depends on the number of subpopulations too. We've seen heterogeneity in a population, so I'm sure there are at least more than 5 subpopulations. Even just simple E. coli. So I think in theory that could be infinite, right? So yeah, so.
And would it be possible, like computationally, for the model to model like 6 combinations of 6?
Oh yeah, easily. Yeah, we can easily do 6 drugs. In our model.
Awesome.
Yeah, it is possible. Yeah, but I don't know if that's enough. So if the question of will it stop evolution, I don't know. I don't think so. I'm sure there'll always be a way to evolve resistance.
Yeah. Thank you. I got 2 questions too. The first one regarding the sequential combined therapy. You applied for heterogeneity in Petri dish, but do you have any plans to apply it to a real in vivo infection, maybe in another model or even in humans? And if so, how would you think it could be done?
Yeah, I mean, I think, for cancer, people are doing these single-cell RNA-seq studies, right? So I think it's getting there for cancer, but bacteria, it's really hard to do single-cell measurements. I mean, single-cell transcriptomics, at least, yeah. So I think people are developing these. Maybe what would happen probably is that, in our study, what we found these subpopulations, we found just 3 enzymes that could explain the 2 subpopulations. So then if you could come up with some fluorescent reporter, right, for those 3 genes, then you can put that in vivo and see if those change. I think that'll be the best way to do this in vivo. Because in vivo, you can't measure all 4,000 genes, but if you use a computational model, we can find those 3 or 4 genes that are most important. Then you can, like fluorescently label them and put them inside a mouse. Then you can see, if they're changing. Then we can look at different subpopulations. Yeah, but that's a good question.
Yeah, thanks. And the second one has a little bit to do with what David said. For example, assume like this model works, and for example, with 3 drugs. I don't know, Acetobacter Baumannii is no longer a problem. But what if Baumannii becomes resistant to that combination? Then should we use now 4 drugs or 5 drugs? Or
yeah, I think what David asked is a very tough question. It's really hard because I think nature will always find a way to evolve, right? Like if you throw 5 drugs, instead of evolving resistance in 1 week, it might take 6 months, but it's going to happen. Yeah. So, but I think cool thing about machine learning is that we can quickly identify new drugs, right? So before, you have to wait for a few months of experiments, and then you will find one. Whereas with machine learning now, maybe with 1-day simulation, you can be like, okay, this bacteria is resistant to these 3 drugs. Let's come up with a completely new 3-drug regimen that this bacteria will be sensitive to. So I think that's I would say the advantage of these tools is that you can't beat evolution, but you can at least, speed things up. You can find the next drug quickly.
Yeah. Thank you.
Yeah.
I'm going to read a question from Zoom. Dr. Martinez asked, is it necessary to develop a new AI tool for cancer treatment combinations?
Yeah, I mean, I guess we can't, because the set of genes are much larger for cancer. I think we probably need to develop a new Indigo model for cancer. Yeah, I mean, we can apply the bacterial model to other bacteria, but for applying it to like fungal systems or cancer, I think we need to develop a new model. But there is plenty of data out there, and people are, even my lab is developing combination models for cancer. But it's not the same model, it's a different model.
Another question? No, I have one question.
Yeah.
I think I agree with what you mentioned about taking into consideration metabolism and the way we test resistance in the lab. Dr. Victor Niset at the University of California in San Diego, he actually showed that when he does a test in a more complex mediums, mimicking or Sera, then bacteria that in theory are super resistant are really sensitive to antibiotics.
Yeah.
So that's the comment. So that agrees with you. Probably some of what we consider resistant, it depends on the type of test we do, right?
Exactly. Yeah.
Okay. Then regarding about the questions that they have been already asked, in this, considering the whole the human body and the organ where the pathogen is, probably also has to do with the timing. Considering the circadian cycle,
that's true.
cycle because that also improves metabolism. Is totally different at night than.
Yeah. That's good. That's a good point. Yeah. I mean, right now we're just saying glucose is there or not there, but it's possible glucose is here in the morning. Something else is in there in the evening, right? Yeah. I think the one direction we want to do is build more dynamic models that that also take into account time and evolution. So I think the caramel model, because it's based on a metabolic model, we can, the next step would be similar dynamics in there. But it is extremely hard to do, how does variation of metabolites, influence the outcome? I think that's a very interesting question. Yeah.
Yeah, and the other thing is that also it's known that immune cells, when they are confronted with pathogens, the metabolism change totally.
Exactly.
And then produce new metabolites that probably the bacteria are using to.
Exactly. Yeah, I think if someone does metabolomics and figures out what metabolites are actually being produced, I think that'd be really cool data we can then feed into our model and then say, okay, maybe those metabolites that immune cells produce, maybe they work in synergy with antibiotics, or maybe we should find antibiotics that actually work in synergy with the metabolites the immune cells produce. I think that would be really cool. That's a good question. Yeah.
Thank you. Another question? Oh, there is one more in Zoom. So. Alberto Pantoja is asking, can we take the different routes of administration of antibiotics into account for what we are discussing? And the second is, how can we know when we are over parameting the model?
Yeah. So I guess the first question, is interesting. I think the last part of the talk where I talked about the pharmacokinetics, that's where we can include the route, whether it's IV or just oral. So I think that's definitely something we want to include in our models, and I think that's possible for which we have data on. And over parameterization is, I think it's sort of I think it's asking about overfitting, right? So it depends on, but we can actually, for the machine learning part, we can figure if the model is over parameterized by sort of hiding some data and then seeing if the model works really well. But the only way to test if it works is generalized as well is, trying it in different hospitals or different regions worldwide. So the last part, I think, was this. Yeah, we've been using data from worldwide places, but it is possible that the model we develop for, China doesn't work in India, or even within India, South India models doesn't work outside. So that's a big challenge. Yeah, and I don't know how we're going to solve it. Maybe we'll build a model separately for South India or Mexico. But yeah, it is. But that's a very hard challenge. Yeah.
Okay, if there is no more questions, thank you very much again for your presentation.
Thank you. Okay, can I ask the final question?
Sorry, I was trying to get my mind up to what I wanted to ask. The thing is, how could we account for mutations already found in highly resistant strains of bacteria in these models, for example?
How do you account for if it's already drug resistant?
Yeah.
Is that, if it's already drug resistant, there are, there are two ways. One is that if you know what genes are mutated, we can include that in our model. We can either turn them off or on depending on what the mutation is. Another is that if you know what drugs the bacteria is resistant to, we can remove those drugs from our model. We can, when you make the predictions, right? Let's say we have 1 million predictions and 500,000 involves drugs that it's already resistant to. We still have we can still look for drugs that it's still sensitive to. So we can find combinations for that the bacteria are susceptible to, sensitive to. So there are two ways we could include that in our model.
Yeah, and like, for example, I wanted to know if considering these machine learning models, if we could find patterns of resistance in like many, not only strains, but species of bacteria, like the ones that are resistant to a certain antibiotic, could be, well, many of those sensitive to a certain combination. I mean, if we could find some patterns there in order to save some time in the case of infections that may be, that may take time if we wanted to screen the genes of the bacteria in the case of clinical repercussions.
Oh, are you saying if we know that a bacteria is sensitive to resistant to certain drugs, can we make predictions on what drugs will be sensitive to?
Yeah.
Yeah. So that's the idea behind this camel, is it sort of takes the time factor into account. Was it long? Oh, okay, many slides back. Yeah, this one, right? Like, so this takes into account the order in which you add the drug. So it's possible that, when you add drug A, first drug, bacteria is resistant, then we can predict the second drug where it will be sensitive to. So that's sort of mimicking what you were saying. The drug bacteria is already resistant to certain drugs. Can predict some drugs that will be sensitive to? Yeah. I mean, ideally, we would want the genome sequence, but roughly you can guess, if it's, at least for E. coli, if it's resistant to what it might be sensitive to later on. Yeah.
Thanks.
Yeah. Thanks.
Thank you very much.
Thank you. Thanks.